graft that's put in pulmonary position. And here you can see the pulmonary autograft, and you can observe this dilatation, where in the left case we reinforced it with a kind of textile, whereas in the right case you can see the unreinforced but really dilated pulmonary artery that's now under systolic, well, diastolic and systolic blood pressure. So I just wanted to show this picture to keep on reminding myself and ourselves as engineers that often there's a very large discrepancy between the nice models and drawings that we have in our mind as numerical analysts and then the surgical reality. Okay, but so what did we do in these animals? So these surgeries were performed, and then we did MRI follow-ups 4 days, 2 months, and 6 months after surgery, so you can observe how the pulmonary autograft dilates further and further in time. And here we put that in a more quantitative way. So what we show are diameters. So the maximal diameter measured from the MRI scan. First for the segment of aorta more proximal to the autograft at 4 days after surgery, at 2 months, and at 6 months after surgery. And then the same for the distal aorta. So you observe a bit of dilatation, which can actually be due to the autograft that's, or the effect of the autograft that's well, let's say diffusing a bit into the distal and proximal end, but also these sheep were still not fully matured. So there's a bit of growth, just autosomal growth that's involved here as well. And here you can see the results of the pulmonary autograft. So you can really observe the significant dilatation and increased growth or further dilatation after the implantation. Whereas if you reinforce with a mesh, we also have this immediate dilatation, but the dilatation is halted after a while, thanks to the mesh support. Okay, so these are just the numbers that we can get from the MRI, but an animal model can only tell us so much when we observe it non-invasively. We really need to dive in and measure more if we want to understand what's happening to all the tissues that are involved. So that brings us to the in vitro analysis of the samples after animal sacrifice and explantation. And well, from this point on, we're only going to focus on those animals without mesh reinforcement. I could also talk about the ones with, but I think there's already enough material to fill up one presentation just with the unreinforced case. So the first in vitro analysis that we did was microstructural. So obviously inside a blood vessel, we have different layers. We have the endothelial layer, the medial layer, and the adventitial layer with all kinds of cells inside, and the elastin lamellae, etc. So we want to, through histology, we did stainings to be able to observe all the constituent contributions, to also quantify them. And we also measured all the thicknesses of the layers and just the thickness in general of the wall. And here are some results, where on the left you can see a native pulmonary artery, so before anything happened to that artery. And then on the right, the remodeled pulmonary autograft after 6 months. And this is a Verhoefs van Gieson staining. So pink shows the collagen, darker purple shows elastin. And what you can see on the bottom image, it's, well, first the top image, there's a lot of extra collagen, and the wall is at least as thick or even thicker than the native pulmonary artery. Whereas in the bottom figure, we don't see a lot of collagen left, and the elastin fibers appear stretched, and there's thinning of the wall. So, and actually these are just 2 examples of histological samples, but we really observed this, well, let's say these 2 groups in all the histological samples that we took. So we will just refer to this, let's say, as type A remodeling and type B remodeling. And this kind of remodeling, so these 2 slices are actually taken from the same sheep and from the same circumference. So some locations showed this kind of type A remodeling, whereas others showed type B remodeling, even in the same animal and at the same circumference, let's say. On the other hand, we performed planar biaxial tests, so mechanical tests on the resected tissue. So again, on the aorta, on the baseline pulmonary artery, and then on the pulmonary autograft that remodeled for 6 months. So we resected the tissues, we make sure that we align them with the natural longitudinal and circumferential direction. We apply a speckle pattern so that when we mount them in our biaxial setup, we can do DIC, digital image correlation, to get a strain map. And so then we load them into our biaxial device and perform the testing. And this mechanical testing, well, we really did a lot of optimization and tweaking into the protocol. We did all of that at Fiber. So that's our KU Leuven core facility for biomechanical experimentation. It's just 4 years old. I'm actually quite proud of it that we were able to bundle all infrastructure and expertise for biomechanical characterization of biological tissue in that facility. And actually, in that context, I just want to make a really small intermezzo on a challenge, a community challenge that we're currently organizing together with the Avicenna Alliance and the VPH Institute, so the Virtual Physiological Human Institute. So this actually came from, I was saying, a lot of optimization and tweaking went into planar biaxial testing, and I think all the experimentalists present in the meeting can probably imagine that, or have already experienced that there is so much variation if you go through literature on how to perform mechanical tests on biological tissue, not only biaxial, but even uniaxial, or even on bone tissue testing. There simply isn't a lot of consensus, let's say. And so we launched this initiative, which we call C4 Bio, so a community challenge towards consensus on characterization of biological tissue. And we decided to start very simple. So we're currently running a test campaign on how to do a simple uniaxial tensile test on aortic tissue. And I didn't want to spend a lot of time on it, but if you're interested, this first campaign is already ongoing, but we really think that this is an initiative that will go further and will move towards other testing campaigns. And there are quite some UK participants, but no one from Sheffield. So if you want to, or if you're interested in the initiative in general, you can go and check out the website and, well, and let us know if you want to be involved somehow. But so that was intermezzo. What we did here was the planar biaxial tensile testing on those different tissues. And before we move to more advanced interpretation of the curves and curve fitting for our numerical models, we first wanted to assess the results just by looking at the slopes of the stress stretch curves. So we just, we will display the results in terms of showing the slope at 20% stretch, at 15% stretch, 10, and so forth, for the circumferential and the longitudinal stress stretch curves, just to have a more quantitative way to interpret all those graphs. So, well, here you see the slope as a function of the stretch level. So here, the first blue bar is at 20% stretch, the slope of the proximal aorta, and here for all the other stretch levels. So you can observe the stiffening of the material, which is as expected. Same for the distal aorta, a bit more compliant distally. And then you see the pulmonary artery, which is even more compliant, well, is as expected more compliant than the distal and proximal aorta. And then we plot the pulmonary autograft after 6 months of remodeling. And just as in the histology, we actually observe two groups. We observe one group that appears to have become rather stiffer, but fails early, fails after 20%, whereas there's another group that appears to, well, to, let's say, keep the more nonlinear stiffening and is able to reach the higher stretch levels. And the same kind of trend is observed in the longitudinal results, so longitudinal slope versus stretch. Well, this is basically a way to try to interpret the massive amount of data that we got. What I'm still very disappointed about is that we were not able to match the location of our mechanical samples to the histological samples. So we can't confirm one-on-one, although we have a hypothesis, of course, which type of remodeling belongs to which histological image. And the reason for that is that we simply had not anticipated before we started the animal experiment that there would be so much inhomogeneity within a single blood vessel. And this is something that really still frustrates me, that we didn't think about it. So another reminder to myself and maybe in general, that when designing your experimental protocols, you really have to try to anticipate the unexpected as much as possible. But sometimes, well, that doesn't work out. Okay, but still, we have all this data. And let's say as a conclusion of our in vitro and animal work, we observe what is also observed clinically, namely some tissue moves towards a homeostatic state after you bring it to this high pressure environment by having what we call adventitial fibrosis, so new collagen that's appearing, whereas other tissues seem to dilate further and they fail. They create this aneurysm kind of appearance. So, well, that's what we observe. But unfortunately, let's say, even in our more controlled animal study where we could measure more, we still see that the loading conditions, or the whole environment is still too inhomogeneous to be able to draw direct conclusions. And just for you to appreciate that, here are the MRI scans of the 6 sheep, so the baseline MRI scans after implantation of the pulmonary autograft. So you can observe immediately the amount of variation that's present in those sheep, even though they are all same age, same strain, and the same surgeon performed the procedure. So even then, there's a lot of variation. So the current loading conditions, let's say, or boundary conditions in general, are too inhomogeneous to draw direct conclusions. So then there's 2 things that we could do, and what we are doing. On the one hand, we're looking to make our conditions even more controlled and more homogeneous. So we're looking at, bioreactor setups where we can, in a more controlled environment, stimulate this arterial tissue in different ways and see how the tissue responds to the different mechanical stimuli. On the other hand, of course, we can go for in silico analysis, and that's what I wanted to show here. And I think, well, many of you are actually numerical analysts, so you know that a lot has to go into a numerical model to be able to make it as reliable as possible. Obviously, the segmented and the mesh geometry, the boundary conditions in terms of blood pressure, but also in terms, in this case, of the axial pre-stretch and how it's attached to the environment. A material description, which, well, we will talk about in quite some detail, because in that material description, we want not only to put information that we got directly from the mechanical tests, but also information from the histology and cellular biological function. So what kind of, let's say, what kind of gene expression is going on in these tissues as they are forced to now adapt to this new mechanical environment? And can we introduce that kind of activity in the material description? And obviously also a good, reliable and stable numerical implementation needs to go, is required to have a good numerical model. So let's first talk a bit about the material description. So we want to describe a pulmonary autograft and an artery and an aorta. So when we talk to biomedical scientists, they of course say, well, you need to have all these biological structures inside. But when you talk to a mechanical engineer, he says, well, this tissue has to cope with some pressure boundary conditions and some displacement boundary conditions. So we need to treat them as this kind of structure of springs that are placed in parallel. So we have to try and merge these 2 worlds. And the way we do that is simply by considering that each parallel spring in our structure, which I now obviously represent in a 1D kind of way, but this is a 3D structure. So every constituent actually is one of the parallel springs in the structure. So we have elastin on the one hand, we have collagen fibers that are embedded, we have smooth muscle cells, and basically we could even put more constituents in or out of the equation depending on how complex we want our material description to be. So that brings us to the quite well-known Gasser-Ogden-Holtz-Apfel representation of material, where we have our strain energy density function. So let's say the potential energy that's trapped inside the material because of the deformation of all those constituents. So here we have the contribution of the elastin fibers, and here we have a nonlinear stiffening contribution of the collagen fibers. And while this strain energy density function, I guess most of you are familiar with it, if it has the property that if you take the partial derivative with respect to the deformation gradient, so the global deformation gradient of such a volume element, then you end up with your stress tensor. But we actually use what we call constrained mixture theory, which was introduced by J. Humphrey and colleagues. So what we do is we add a bit of differentiation in that strain energy density function in terms of having a different deformation gradient for each of the constituents. So elastin can have a history of deformation going from birth, actually, or from the maturing, or let's say all the elastin that is present in your body is said, or in the artery is said to have actually grown along with you from when you were born, whereas the collagen is continuously turning over. So the history of collagen is far shorter, and there's less deformation that this collagen has experienced when you look at it in time. And also we can allow the fractions of elastin and collagen to change. So each constituent can have its own turnover rate and stress-free configuration, so its own deformation gradient. Which here, I'm not showing the deformation gradient, it's the invariant, or one of the invariants of this deformation gradient. And what I just wanted to add is the smooth muscle cells are there, but they don't contribute mechanically. They are there as sensors, but they don't have, or in this equation at least, we didn't attribute extra mechanical stiffness to these smooth muscle cells. So what do we do in this strain energy density function? We have some extra partial differential equations to govern the disappearance and the appearance of all of those constituents. And in our experiment, we assume, these animals are almost fully grown, so we assume that there's no extra production of elastin. And because it's a relatively short time span, just 6 months, we assume that no significant elastin degradation takes place. This is definitely a big assumption that's under debate and that we should probably improve upon in the very near future. But that's what we assumed in the models that are to come. On the other hand, we assume that the smooth muscle cells, they don't contribute mechanically, but they are there as sensors, and they sense what the extracellular matrix is feeling in terms of stretch. And if they feel a stretch that is deviating from their homeostatic stretch level, they can start to differentiate from a contractile phenotype to a synthetic phenotype, and they can also start to proliferate. And so what I mean with a synthetic phenotype is that then these are the cells that actually start to produce collagen. So, for that we need to obviously establish what the homeostatic stretch level of smooth muscle cells is, and also a rate parameter that will govern how quickly they will switch their phenotype and how quickly they will proliferate. And then, like I said, so the smooth muscle cells, the synthetic ones, they can produce collagen, and they will actually do that again, enhanced by the non-homeostatic conditions of the ECM. So the more the stretch in the smooth muscle cells deviates from what they are used to or what they want to feel, they will actually start to produce more collagen, again, with a certain rate parameter. And on the other hand, collagen is there, but it's actually also continuously degrading, which again can be enhanced by non-homeostatic conditions. So if the collagen fibers feel, in this case, it's an energy-based criterion, if they feel too much strain energy, the degradation process will be expedited. So these are extra equations that we plug into this constrained mixture model that I already showed you. And in a first instance, we don't yet go to more complex finite element implementations, but we just, in a very simple 1D setting, we test our material description, and we virtually test this hypothesis that I'm going to explain. So we have a kind of type A material in which the smooth muscle cells are really sensitive to their environment, and they, as soon as they feel an increased stretch, they will immediately and very massively differentiate into synthetic smooth muscle cells, and they will start to produce collagen as quickly as they can, let's say. Whereas in the type B, we hypothesize that if there is too much stretch, the smooth muscle cells lose their connection to the extracellular matrix, and they just do what they were always programmed to do, and they uphold a constant turnover of collagen as if nothing was changed with respect to their initial situation. So these are two types of remodeling that we tested in a virtual 1D situation. We did that in MATLAB. So we have an artery that's used to pulmonary pressure, and it's in homeostatic conditions. And then we bring that artery to non-homeostasis. So we apply aortic pressure, and on the one hand, we simulate what happens in this type A remodeling, so fast response, high sensitivity, and then the type B remodeling, slow response and low sensitivity. And then if we plot the results in terms of collagen density and in terms of radius, we can really observe the same kind of remodeling, or at least qualitatively the same kind of response as what we observed in the histological study. So in the one case, a thickening of the wall, and in the other case, a further dilatation. The nice thing of virtual models is that you can also do virtual biaxial testing. So you can extract virtually a piece of tissue from that remodeled sample. You can perform a virtual biaxial test, and then also, in this case, we can correlate the mechanical response to what we could call it the histological or the morphological outcome. So that's it with respect to material description. Then the next part that we need, of course, is computational resources and a reliable and stable numerical implementation. And I won't go into those details, but I just wanted to show this publication that we made on UMATs, because we actually wanted to share all the efforts that we put in terms of trying to get a fiber-reinforced hyperelastic material implemented in Abacus, in our case, because it really led to a lot of frustrations and errors that we did not expect, and we think that other people will also not expect. So we decided to publish, let's say, all the struggles that we went through, and the code in terms of the UMAT is also public on our GitHub repository. So feel free to go and have a look at that if you would like. Okay, so that means that we have everything we need to perform our simulations. So what do? We have our geometry of a certain sheep. We have the material parameters exactly from the mechanical test. So the baseline pulmonary artery and the baseline distal and proximal aorta. So we can plug all those into our material model, and we can allow it to virtually grow and remodel for 6 months. And the nice thing, of course, of virtual testing or virtual clinical or animal trials is that you can do that in a far shorter time span. So we can do that in 180 days, can be simulated in less than an hour. And these are just some basic results. So here you can see for 2 sheep, the experimental values in terms of diameter and the model values in terms of diameter. So let's say we're able to capture this dilatation, at least from a qualitative perspective. And we can track the diameter, but what's more interesting and what makes these models less phenomenological and at least a bit structurally and mechanobiologically inspired is that we can also track other things that we can compare to our experimental data, like collagen fraction. So we can really see how the fraction of collagen evolves in time and how that also differs in different locations of the structure. And we can do virtual biaxial tests on different locations in the same animal. So there we can try to observe exactly what happens in this case, the bulging side, so the side that was more stretched, and the non-bulging side, the side that was less stretched in this particular system. And you can really see and then observe how the material properties change in time. So other than in an animal experiment where you can only intervene at the end, here we can continuously monitor how the properties are changing in time. Which again, well, we can observe that in the non-bulging side, a more kind of type A remodeling is taking place, whereas in the bulging side, the dilatation is so large that the system cannot cope, or the smooth muscle cells cannot cope, or at least that's our hypothesis, which seems to match well with experimental reality. So that's, let's say, the basic idea of our models, and we're far from ready yet, but the question is, at least already, if we would have this model completely up to date, could we use it now to adapt treatment to avoid failure? And that just brings me back to one of the first slides. The idea, of course, is that you would be able to, with the preoperative data of an actual patient, with also preoperative material properties, you could simulate and play around with those design parameters and steer the design parameters or optimize them such that you would move towards the type B kind of remodeling rather than the aneurysmal remodeling. Okay, so that's let's say the ideal world, but we're not there yet. There's still a lot of room for improvement. One of the most important things that we think should be included is the effect of the fluid flow, and the fact that this fluid flow is definitely disturbed in this kind of aneurysmal situation. So what we're currently working on is our MRI data also included flow analysis. So we have flow information, so we can calculate through fluid structure interaction, we can calculate the wall shear stresses that are present, and we want to include wall shear stress as an extra driver for remodeling, because we still observe that, let's say, experimentally, there is still more inhomogeneity than what we can get from our models right now. And we definitely think that this wall shear stress and the inhomogeneity in that wall shear stress is an important driver and important factor for this inhomogeneous remodeling. So that's one big step for improvement, but basically all of the aspects that I already mentioned have a lot of room for improvement still there, with one very important one, that everything is currently still deterministic, whereas of course there is a lot of uncertainty on all the parameters that go into this kind of model. So non-deterministic modeling is definitely necessary if we want to really go towards a kind of predictive tool, something that we can present to a surgeon with margins of probability. Of course, we will only be able to predict a certain outcome with a certain confidence, within a certain confidence interval. All right, so that brings me to the conclusion of my talk. And well, obviously, I definitely didn't do any of this stuff all by myself or alone. There were a lot of people involved in this project. Definitely also the people from cardiac surgery who brought well, all the clinical know-how and all the biomedical know-how into it. Also molecular and vascular biology. A lot of effort going into the medical imaging, and then also our biomechanics group with also people from the University of Ghent collaborating for the fluid structure interaction modeling. All right, so that was my talk, and I'm happy to answer any questions if you would like.
Thank you very much, Neil. That was a fantastic, really clear talk, and a really, in-depth look, comprehensive look from different views in this ROS procedure. I learned a lot about. I'm sure all the experimentalist clinicians and modelers, you made everybody happy in this in the audience. I'm sure there'll be lots of questions. So everybody, anybody if you have any questions, please put it in the chat box, and I will invite you to ask it directly to Nela. In the meantime, I have let's say a question. So you showed in the very beginning that there are different methods of different interventions that are available, of which Ross procedure is one. So Ross procedure seems to have the very high performance compared to the others. Now the other interventions possibly fail for different reasons. Is it possible to learn something from them or their biomechanical competence and see if they can be that learning can be used to improve the performance of the Ross procedure itself?
Yeah, I think that's a very good point. And indeed, I've sort of centered my talk around the Ross procedure, but I think the question that we're trying to answer is broader than that, in terms of we're trying to quantify and understand how tissues respond to an altered mechanical environment, and that can happen in many situations. So not only in an aortic valve replacement, but also in aneurysm formation in general. But it's a very broad question, and we wanted to narrow it down to, let's say, a baseline being a healthy tissue, because of course you have aneurysm formation or a diseased situation when you have an aortic valve replacement, whereas in the Ross procedure we at least assume that the pulmonary tissue is healthy, and we want to see how this healthy tissue remodels. But that's just like the first part of the question, because then you want to see how much of that translates into well, thoracic aneurysm or abdominal aneurysm remodeling. And well, obviously there's a lot of research going on there as well, and I think there can be a big interplay, and we can learn from each other there.
Thank you, thank you. Next question is from Professor Damien Lacroix. Damien, I have made you presenter, so please go ahead.
Yes, thank you very much. It was a really nice presentation, really enjoyed that. So I had two questions. The first one was, so in your fiber reinforced hyperelastic model, I think that there's no time dependency, so you basically have a given strain rate, which is the one that you've used probably from your bioreactor testing, and that, so your results might be very dependent on actually the strain rate, but also if you've got some small macro changes within the tissue, now the strain rate, local strain rate, might also change quite significantly. So I was wondering whether you thought, I don't know, whether this was a limitation or you'd think about a different approach.
Yes, so indeed, it's thank you for the question. It's a hyperelastic model, but the growth and remodeling model that is implemented makes it or makes it equivalent to a kind of viscoelastic model. So the time dependency, but that's on a different time scale, is there in terms of the tissue responding and then remodeling, and you could see this remodeling response as a kind of viscoelastic term. But you're absolutely right that, well, the hyperelasticity baseline disregards any time-dependent effects in our tests. So the way we do our biaxial tests is we apply the same strain rate as physiological, as what is there physiologically. So we use the strain rates that are equivalent to, let's say, the movements between diastole and systole in the body.
Yeah,
and we disregard.
If locally, I was just wondering whether, if you have some small changes in the aorta, then locally you might actually have some very, sudden change in strain rate on the tissue.
Yeah, I think yes, and especially I think in the fluid structure interaction model shows that, and that indeed will become more important as soon as we include that, because at this point we just included let's say a uniform pressure, and we just actually simulate even not a varying pressure, but just an time averaged pressure. So so that hasn't come into play yet, and you're absolutely right that we need to include that, and that dependency will indeed induce more inhomogeneity. I fully agree. Yeah.
And my second question is a bit more general, but I was thinking, so some of the major problems are relating to toxin environment that is happening within the tissue. So whether somebody who's smoking, or there's chemical toxins in the environment, based on pollution, or everything, and that's what induces damage to the tissue. And I'm just wondering how your approach could include that. What would you do to what would you do to modify this to be able to include this?
I think it's a very good point. And well, apart from toxins, inflammation is also very important. So especially here, well, there's a surgery, and a big part of the remodeling, although we didn't include that here, but that's something that we are including now, is not due to the mechanical overloading, but it's due to the fact that there's a large inflammatory response going on. And well, let's say the toxins could have a similar kind of effect, so a non-mechanical driver to the remodeling. But I think, let's say the model and the way it's implemented it's of course it's still relatively phenomenological, and it's homogenized at the level of well, let's say at the continuum level still, but we can simply add an extra differential equation that brings the toxins in, or that brings the inflammation in. But of course, every extra equation brings extra parameters, and that's something that will always be a challenge to do curve fitting on, or to obtain. But it's definitely a very important factor. I agree.
Thank you.
Thank you, Damian. I think the next comment is from Professor Paul Evans. Paul, I've made you presenters. Please go ahead.
Oh, yeah. Thank you, Nella. And the first about Paul Watton. I don't think he's on the call, but Paul does some very nice work looking at shear stress and modelling of aneurysms in Sheffield.
Yeah.
So, do you know his work?
Yeah, I'm familiar with his work. I spent some time during my PhD in Gerard Holzapfel's lab, actually right at the time when Paul Watton was also there. Okay. So we met then. Well, I was a small PhD student at the time. I don't know if he remembers me, but I definitely remember him. Yeah.
I just wanted to mention that as he's not there, and like worth reaching out to him. And then I just picked up on this comment about inflammation you made. So you're absolutely right. In vascular surgery, there is inflammation, and it's quite acute, and it can cause damage, but then it subsides. And I'm just wondering whether in your sheet model you could, in that external sheath that you put on, you could immerse it in glucocorticoids or a steroid to try to limit that initial inflammatory response, and that might be quite interesting.
Yeah, it's very nice that you bring it up, and indeed that's something that we're trying to look at. So we have a collaboration now with a group who is making these through electrospinning can create or do additive manufacturing to get these kind of structures, and they have a kind of coaxial so the wire that they melt or spin is coaxial, and it can release indeed these kind of anti-inflammatory agents. But well, let's say it's so preliminary. We have no idea yet how much and when, and so there's just so much to learn still. But I think indeed that's the future. And to have a kind of response, to have an external support that might be bioresorbable, because you want to support the artery in remodeling, but once it's remodeled, you don't want any foreign body there anymore, ideally. So you would have a material that resorbs at exactly the right rate. But I guess there's so much engineering still left there to try and figure out exactly how that should work, and on a patient-specific level even.
Thank you.
Thank you for the remark.
Thank you, Paul. If you have any questions in the meantime, please feel free to add them in the comment box. Nela, I had yet another question. Sorry for pestering you. So the question is really, I know you have to go for another meeting after this, so I'll keep it short. So the MRI images that you capture in the sheep, could they potentially be used to discover any biomarkers of what might happen later on to the tissue, I mean, later in time?
Yeah, of course the resolution of the MRI is still not the way we would like it to be. So at this point we can only see the lumen, so we don't see anything related to the wall. If anyone has ideas or better scanners so that we could actually really observe what's going on in the wall, that would be great. We do ultrasound, but of course, I mean, depending on, sometimes we also have surgeries where we put it in the carotid, and then with ultrasound you can already see something because it's quite superficial. But here, because it's rather deep in the chest, yeah, the MRI is all we can get at this point is the lumen. So with flow, but and so the overall diameter, but not enough to learn more about the tissue itself, unfortunately.
Great. Okay. Well, something perhaps will develop over time with our technologies developing.
Yes,
great. Okay.
Looking forward. Let me know when you have it.
Well, not me. Not me. But there is another question. Paul, do you want to comment? Sorry, please come to the microphone.
Yeah, I think I saw the question. So IVUS would indeed be a very nice alternative as well. Yeah.
Yeah. With the virtual histology, you could get that structure.
Yeah.
But of course, you've got to persuade somebody in the hospital in Ghent to allow you to take sheep in and into the cath lab. So that might be a practical consideration.
Yeah, we already have a nice animal surgical facility at UZ Leuven, but yeah, the more expensive equipment that stays reserved for the patient. So it's always a bit of a fight to get the equipment in the facilities. But it would be nice.
Super. Thank you very much, Nela, and really appreciate you coming and giving us a talk. It was really enjoyable for all of us. Hopefully, we'll see you here someday in person. And
nice. Thank you for having me.
Welcome. I'm sure everybody enjoyed here as well. And so here's wishing everyone, and Nela, of course, wants a big hand to you, and have a nice holidays and a restful break.
Great. Same for you. Stay safe. Bye-bye.
Bye. Paul, did you want to say something? Sorry. I see a raised hand from Paul. Did you want to say something, Paul?
No, that was just from before. Bye-bye.
All right. Okay. Bye.
Thanks.
Bye.
Thanks, both of you.
Bye.